Good morning, everyone, and welcome. I am delighted to have you here today. My name is Dr. S.K. Singh, and I hold a PhD in this field. Today, I will be discussing concurrent validation in sterile manufacturing. This is an important topic that ensures the quality and safety of our sterile products. Over the course of this presentation, we will explore the key concepts, procedures, and best practices involved in concurrent validation. I'm excited to share this knowledge with you, so let's get started. Today, I want to talk about validation that is performed simultaneously with commercial batch manufacturing. This approach is particularly useful when product supply is critical, or when we don't have a long manufacturing history to rely on. By integrating validation into the actual production process, we can ensure that everything is validated in real time, which helps us monitor process consistency and sterility assurance directly as the product is being made. It's a very proactive way to maintain quality and compliance, especially under tight timelines or limited historical data. Of course, it's essential that we follow strict guidelines, such as GMP, Annex 1, and other regulatory expectations. These standards help us ensure that our validation process is robust, consistent, and compliant with industry regulations. Overall, concurrent validation is a powerful tool to enhance our quality assurance, especially when rapid product delivery and regulatory compliance are top priorities. Let's take a closer look at some of the key factors that influence when we use concurrent validation. First, there's an urgent market demand for these life-saving products, which means we often need to act quickly to meet patient needs. In these situations, there's typically no alternative for patient supply, so ensuring product availability becomes critical. Since the process modifications involved are usually minor and don't impact sterility, we can consider concurrent validation without compromising quality. Additionally, although our manufacturing history might be limited, we do have valuable process knowledge that supports validation efforts. We generally consider concurrent validation when these specific conditions are met, particularly when speed is essential and the risk of process changes is minimal. It's a practical approach that balances the urgency of patient needs with maintaining product quality and safety. Let's take a closer look at some key regulatory references for sterile product manufacturing. First, we have the EU GMP Annex 1, which specifically focuses on the manufacture of sterile products. This guideline emphasizes the importance of maintaining sterility assurance throughout the process. Then, there's the WHO TRS 937, which offers global standards and best practices for sterile manufacturing, helping ensure consistency and safety worldwide. Moving to the US, the FDA guidance on process validation is crucial. It highlights the importance of validating all processes to consistently produce sterile products of the highest quality. Additionally, we have the PIC-S Annex 1, which aligns closely with EU standards, but also incorporates international best practices for aseptic manufacturing. Across all these references, several core requirements stand out. First, sterility assurance is paramount. We need to implement robust controls to prevent contamination. Next, environmental control plays a critical role. This means managing cleanroom conditions, airflow, and particulate levels diligently. Lastly, aseptic process reproducibility is essential. Our processes must be consistently reliable, ensuring each batch meets the same high standards without variation. These key points form the backbone of regulatory expectations and help us uphold the integrity and safety of sterile products in our manufacturing practices. Now, let's dive into the importance of having a strong prior process understanding. This means we start with a clear grasp of our quality target product profile, or QTPP, which outlines the desired product characteristics. Alongside that, we define critical quality attributes, or CQAs, that are essential to ensure product quality. We also identify critical material attributes, CMAs, and critical process parameters, CPPs, to understand what needs to be controlled throughout manufacturing. Maintaining controlled aseptic practices is crucial, especially when working with sterile products. Consistently following proper aseptic techniques helps us prevent contamination and maintain product integrity. Real-time monitoring and documentation play a vital role here. By continuously monitoring processes and recording data as we go, we can quickly catch any deviations and address them promptly, ensuring consistency and compliance. During commercial batches, enhanced quality assurance oversight adds an extra layer of confidence. This means more rigorous checks and supervision to confirm that everything aligns with our quality standards. Finally, all these elements come together in what's called concurrent validation. This approach allows us to validate processes as they are being performed, rather than waiting until the end. It's a proactive way to ensure our manufacturing process is robust, reliable, and ready for commercial scale. Together, these points highlight a comprehensive strategy to maintain high-quality manufacturing standards and support continuous process improvement. Let's now take a closer look at aseptic gowning qualification. This is a critical process to ensure that personnel are properly trained and certified to work in sterile environments, minimizing contamination risks. Next, we have media fill simulation, which requires prior approval. This is essentially a practical test where we simulate filling processes using a culture media instead of the actual product, allowing us to validate that our aseptic techniques are effective without risking product contamination. Bioburden control is another key aspect. It involves monitoring and minimizing the microbial load on products and surfaces, which is vital for maintaining sterility throughout manufacturing. Environmental monitoring, or EM, is also essential. It involves regularly checking the air, surfaces, and personnel to detect any microbial presence that could compromise the integrity of our sterile environment. HEPA and airflow validation are crucial as well. They ensure that our filtration systems and airflow patterns are functioning correctly, effectively reducing airborne contaminants and maintaining a sterile environment. Equipment sterilization cycle validation confirms that our sterilization processes are consistent and effective. This validation guarantees that all equipment used in production is properly sterilized before use. Container closure integrity testing is another important point. It verifies that our packaging systems maintain a hermetic seal, protecting the product from contamination after sterilization. Finally, all these elements contribute to our overall sterility assurance considerations. They form a comprehensive approach to ensure that every step in our process maintains the highest standards of sterility and product safety. Connecting all these points helps us understand that maintaining a sterile environment isn't just about one or two measures. It's an integrated system designed to safeguard our products from start to finish. Let's walk through the key steps involved in validating our sterile process. First, we start with the preparation of sterile raw materials. Ensuring these materials are sterile is crucial because they form the foundation of a safe and effective final product. Next, we move on to solution compounding and filtration. This step involves carefully mixing the ingredients and filtering them to remove any potential contaminants, which is vital for maintaining sterility. If applicable, we then perform terminal sterilization, which is a final sterilization process applied to the finished product, providing an added layer of assurance. Following that, we have aseptic filling, where the sterile solution is filled into containers under aseptic conditions to prevent any contamination. In cases where lyophilization or freeze-drying is necessary, that process is carried out next. This step helps improve the stability and shelf life of the product. Finally, the product undergoes visual inspection and final packaging. This step ensures that the product looks correct, is free of particulates or defects, and is properly sealed for distribution. All these steps are critical to ensure the overall integrity and safety of the final product, and validating each step is essential for compliance and quality assurance. Now, let's take a closer look at the critical quality attributes, or CQAs, that are essential for ensuring product quality and safety. First, sterility is absolutely crucial. we need to make sure that our products are free from any microbial contamination. Next, particulate matter must be minimized because particles can pose risks to patients or interfere with the product's effectiveness. Endotoxin limits are also vital. These are bacterial toxins that must be kept within safe levels to prevent adverse reactions. Fill volume accuracy is another key factor. Precise filling ensures each dose is consistent and reliable. Lastly, container closure integrity is critical because it guarantees that the product remains sterile and protected from external contaminants throughout its shelf life. Moving on to critical process parameters, or CPPs, these are the variables we control during manufacturing to maintain those CQAs. Filling speed, for example, must be carefully managed to ensure consistent fill volumes without introducing contamination. The force used to insert stoppers is also important. Too much or too little can compromise the seal or cause damage. Filter integrity is another key parameter. It ensures that our sterilizing filters are functioning correctly and not allowing microbes or particles to pass through. Temperature and time during sterilization cycles need to be precisely controlled to achieve effective sterilization without damaging the product. Lastly, differential pressure and airflow are monitored to ensure the environment remains within specified conditions, preventing contamination and maintaining process control. All these critical parameters and controls are interconnected. They work together to ensure that our CQAs are consistently met, ultimately supporting the overall quality and safety of our product. This tight control is what allows us to deliver reliable, safe medicines to patients. Now, let's take a closer look at some key aspects of our manufacturing process. First, we have real-time batch records, which allow us to monitor each batch as it's being produced, ensuring immediate visibility into the process. Moving on, in-process controls are crucial because they help us verify that each step meets quality standards before moving forward. When deviations occur, we have a structured deviation handling system in place to identify, document, and resolve issues promptly, minimizing impact on the final product. Real-time reports are another important aspect, providing instant electronic data collection and analysis, which supports faster decision-making. QA verification for each step ensures that quality checks are integrated throughout, not just at the end, maintaining high standards throughout production. Additionally, we generate a comprehensive summary and evaluation report for each batch, capturing what went well, what could be improved, and lessons learned. All of these activities are supported by strict data and documentation requirements, which are fundamental for traceability, compliance, and continuous improvement. By integrating these elements, we strengthen our overall manufacturing integrity and ensure we consistently deliver safe, high-quality products. Today, I want to highlight that all critical quality attributes, or CQAs, have been maintained within their predefined limits, which is essential for ensuring product quality and consistency. We also achieved a major milestone. There were no sterility test failures, confirming the sterility of our product. Additionally, our electron microscopy trending has been successful, showing no concerning deviations over time. Importantly, there have been no major aseptic interventions needed, which indicates the robustness of our aseptic process. All critical process parameters have remained within their validated ranges, reinforcing that our process controls are effective. The key takeaway here is that these points collectively demonstrate that our process consistently meets our acceptance criteria, ensuring product safety, quality, and compliance throughout production. This consistency is vital as we move forward in maintaining the highest standards in our manufacturing practices. Supports uninterrupted product supply. When we implement concurrent validation, we ensure that the production process keeps running smoothly without delays or interruptions. This is critical, especially in industries like pharmaceuticals, where continuous supply can be a matter of life and death. Real-time confirmation of process capability is another key advantage. Instead of waiting until the end of a validation cycle, we get immediate insights into how well our process is performing. This allows us to make quick adjustments if needed, improving overall quality and efficiency. faster validation timeline is a significant benefit as well. Traditional validation methods can take a long time, but with concurrent validation, we shorten this process considerably. This means faster product release and a quicker response to market demands. And of course, this approach is particularly suitable for life-saving drugs. Because these medicines are often critical, ensuring their production is validated quickly and reliably can make a real difference in patient outcomes. Overall, the advantages of concurrent validation align perfectly with our goal of maintaining high quality, efficiency, and patient safety throughout the product lifecycle. Let's talk about product batches and the review process. They are only released after a full review, which is essential to ensure quality and safety. This process is especially important because of the higher regulatory scrutiny we face today. It's worth noting, though, that this approach isn't suitable for high-risk products unless we have strong prior knowledge of them. In those cases, we need to implement robust risk mitigation strategies to manage potential issues effectively. Now, it's important to remember the limitations and risks involved. While thorough review processes are vital, they can also introduce delays and increase costs. And for high-risk products, without sufficient prior knowledge, the process becomes even more complex and challenging. So, understanding these points helps us make better decisions in product development and compliance management. Now, let's move on to the final validation report. This report summarizes our trend analysis for equipment monitoring, sterility, and failure rates. It's essential because it helps us identify any patterns or deviations that might need further attention. Next, we update our SOPs, BMRs, and control strategies based on the latest data and insights. This ensures our processes remain robust, compliant, and aligned with the current understanding of the process performance. We also have a plan for continued process verification, or CPV, which is crucial for maintaining process control over time. It involves ongoing monitoring to confirm that the process stays within established parameters and continues to produce quality products. Finally, after validation, we engage in post-validation activities. These include reviewing all findings, implementing any necessary corrective actions, and ensuring documentation is complete and accurate. This comprehensive approach helps us sustain process excellence and compliance throughout the product lifecycle. Concurrent validation is absolutely vital in sterile manufacturing, especially when patient supply cannot be compromised. It provides a real-time, scientific assurance that our processes are consistently producing sterile, high-quality products. This approach helps us verify that everything is functioning correctly without delay, which is crucial for patient safety. By ensuring sterility and quality through concurrent validation, we also demonstrate compliance with strict global regulations. In conclusion, this process is a key part of maintaining trust in our manufacturing practices and ensuring that we meet the highest standards for patient care. For further support, please don't hesitate to reach out. We're here to help you implement what you've learned today and to answer any questions you may have. Remember, your success is our priority, so whether it's additional resources or personalized guidance, we're just a contact away. Thank you so much for your time and engagement throughout this presentation. We appreciate your dedication and look forward to supporting you moving forward.